Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about a disease which is usually known as rabies disease. Let me write rabies disease. Now, what are rabies disease? It is a viral disease which is usually caused by, we call that rabies virus. So let me write the point, viral disease. Now, what are the rabies viruses? These are the enveloped viruses, and let me write these are what viruses? Enveloped. They have envelope which is made up of lipid bilayer, and they have a single-stranded RNA genome. Okay, negative single-stranded RNA genome. Now, moreover, these viruses can cause encephalitis. Encephalitis. Now, what are the encephalitis? Encephalitis means inflammation of the brain. Brain inflammation. This virus is usually, transmitted from bat. Then it is, if we talked about that, how this viruses comes, usually the main cases of these viruses are, arises from the Asias and Africas. So in these countries, there are a lot of death occur. So we can say that it is usually transmitted from these countries. So usually the death is been occur in Asia countries, Asia and Africa, and India is a highest rate according to rabies. So we can say India is at highest risk, because in India there is a lot of a rabid dog are present. So we can say that inside the India there is a lot of rabies viruses, and a lot of people is affected from the rabies viruses. Moreover, these rabies viruses, if we talked about the transmission, so first of all, these rabies viruses are found in the bat. Okay, so first of all, it was found in the bat, and after that, these rabies viruses are transmitted to the fox body. So after some time, it is transmitted to the fox body. Now, how it is transmitted to the fox body? This bat was die due to this rabies disease, and after that, this bat was, means that this bat was fall on the earth, and then the fox eat it. So when the fox eat it, so the rabies virus is transmitted through their body, and then this fox meat, or they can buy, or they bite. We can say. This fox bite what? They bite dogs. So then it is transmitted to the dogs. Okay. Usually this, we can say that usually this rabies disease was present in the environment of Asia, Africa and India. It is present. But they directly, affect the bed. First of all, it is these rabies viruses affect the bed. And after that, it is transmitted to the other animals. And as all of you better know, dogs use many peoples, and many of the countries inside many countries, the people use the dogs as a pet. So after some time, it is transmitted to the human. So we can say rabies is actually a zoonotic disease also. So due to each points, it shows that it is zoonotic disease. Okay, now as we talked that it can cause encephalitis, brain inflammation. So usually it is also a neuropathic disease. It is the neuropathic virus. Neuropathic virus. Okay, now it is neuropathic virus. Moreover, if we talked about their genome, So usually we talk that they have a single-stranded genome, okay, negative single-stranded genome. And what actually this negative single-stranded genome means? They means that they can first release, this is negative single-stranded RNA. First they release the positive mRNA, okay, and after that this positive mRNA are now translated, or we can say translation are being started from this positive mRNA, because all of you better know that negative mRNA not directly synthesize the protein. They can synthesize indirectly protein, negative sense, and positive mRNA can synthesize protein directly. Okay, so if the viruses have the negative single-stranded RNA, so they first make the positive mRNA, and then we get the translation. Okay, so that's about the genome. And moreover, rabies viruses have 5 different kinds of genes. Okay, we can say that in single-stranded RNA they have 5 different kinds of genes. So 5 genes are present in their genetic material, or in the negative sense RNA of the rabies viruses. And these are the, let me write, M protein, which we called as matrix protein. They have another protein which we usually called as N protein, which is usually known as nucleoprotein, which can make the nucleocapsid. It can make the matrix. And we have another protein, means this mRNA can code another protein we called as G protein, or we also call that glycoprotein. They can also code for P protein, which we also called as phosphoproteins. Okay, and last we have another protein we called as L protein. L protein is also called as RNA polymerase protein. Okay, so it is the RNA polymerase protein. Now these are the proteins which are present in the form of genes in the negative mRNA. So whenever it is translated, so they can produce each of the protein which are usually necessary for the rabies viruses formation or assembly. Now then we have, so they related with family of Rabidoviridia. Moreover, their species of genus is Lyssavirus. Species Lyssavirus. And the order of these viruses are order mononegavirals. Okay. Moreover, if we talked about the rabies, so usually this rabies is what? This rabies is also, from the start, I forget one thing about the rabies. This rabies is usually, taken from the Latin word. It is taken from the Latin word, which means madness or rage. Okay? Madness. Now that's about the rabies viruses. Then we have the structure of the rabies viruses, that how the rabies viruses will be look like. So let me erase this one and let me explain the structure. Then we have the structure of the rabies viruses. Now how the rabies viruses look like? Rabies viruses usually look like in a bullet-like shape. So if I draw the structure, so it will be look like in this form. Okay, so bullet-like in shape. Okay, they have, as we talked, that these viruses are the envelope viruses. Their envelope is made up of lipid, and moreover, they have, we already talked that they have single-stranded RNA, negative single-stranded RNA, and they have helical proteins. Okay, they are means they are helical in shape. So this is negative single stranded RNA. Okay. Moreover, we talk the length, so their length is 180 mm and their width is 75 mm. Okay, these rabies viruses are transmitted to the central nervous system and cause encephalitis, or we can say the speed of these viruses when through which it is transmitted to the brain. So they are transmitted through a speed of 15 to 100 millimeter per day. So per day they are transmitted to the nervous system, central nervous system, and cause encephalitis. Okay, now this is a little bit about the structure. Now if we talked about the epidemiology of the rabies viruses, as we talked that the main cases of the rabies was, first of all, it was arises in the Asia countries and Africa countries. So many deaths have been occurred at that countries. And we also talked that in India there is India is at highest rate in rabies viruses, because there's a lot of dogs are present in India, so we can say it is at a high rate. Rabies are at a high rate at that country. Moreover, first of all, rabies means the Switzerland can protect, or we can say Switzerland can protect themselves from the rabies viruses. How they protect the rabies viruses themselves? They can take the chicken heads. So first of all, they take the head of chicken, or they vaccinated this head of chicken with a live inactivated vaccine. Now after some time, with this combination, chicken head and live inactivated vaccination, with this combination, then they allowed the fox as well as they also allow the dogs. So then the dogs and fox were allowed. Dogs and fox were allowed to eat this head of chicken which is vaccinated. Now once they eat up, so after some time their body is vaccinated. So a vaccine means was prepared, and moreover the immunity is developed inside each body, and finally the dogs were protected from the rabies viruses. So we can say that we can also vaccinate our pets as well. Okay, now this is how the rabies viruses, we can say that it was eliminated from the Switzerland, but somehow it is eliminated. Then we have in 1997 to 208 1997 to 2008, this rabies viruses was fully eliminated, or we can say 98% it is eliminated. Okay, so 98% it is eliminated from the Italy. Then we will see the pathogenesis of the rabies viruses. Okay, so let me write pathogenesis. These rabies viruses usually, how they can start their life cycle. If we talked about the life cycle, so we already talked that it is usually, first of all, it was present in the bat. Then bat was eaten by the dog, means by fox. Then dog was bitten by the fox. Then rabies viruses are transmitted to their body, and due to this we call that zoonotic disease. Moreover, we use dog as a pet. We can take it at our home. We use it for different purposes, for the security purposes and some other purposes. Okay. Moreover, this dog once bite the human body, means this usually this rabies was present in the rabid dog. So let's say we have the human body. At this case, let's say this is the human body. This is not the exact of the structure of the human body, but it is just an example. And I'm really sorry for my beautiful diagram. So let's say this is the human body. It was bite. It was bitten by the dog, and we called it rabid dog. Now once it was bitten by a rabid dog, then what actually happened? At the site where it was bitten, let's suppose it was bitten at the leg portion, then the rabies viruses are divided here or replicated here, but not a lot. Just a few rabies viruses are divided at that position, and then it is transmitted to the peripheral nervous system. Okay, once it was get matured, then it was transmitted to the peripheral nervous system, and after the peripheral nervous system, it is transmitted to the, so this was, let's suppose, the peripheral nervous system, and after that it was transmitted to the central nervous system, and from central nervous system they can start their replication processes. But how? They first of all find the neurons. So all of you better know the neuron structure is just like in this form. So let's say this is the neuron. Now they find the neurons and attach their self on it, because there was the receptors of the neurons. There was the receptors present on the neurons, which are which are usually matched by the rabies viruses. So rabies viruses, let's say this is the rabies viruses, as we talked that it is bullet in shape. They attached here, so attachment is done. After that it is engulfed, or it is internalized. We cannot engulf it. It is usually endocytosed inside each cell, and once it is endocytosed, so then they fused with the lysosomes, and after that they release their genetic material. Let's say they release their genetic material, which we call that negative mRNA. Now once they release their genetic material, so it is this mRNA is used as a template to produce positive mRNA. So after that we get positive mRNA. Now after positive mRNA, they produce two things: more copies of the negative mRNA. So we get more copies of the negative mRNA, and moreover this positive mRNA can also release the structural proteins, as we discussed in our genome. Okay, we already discussed that what kind of structural proteins: M protein, P protein, nucleoproteins, all the L protein, all these are the structural proteins. Structural proteins. Now once they release the structural proteins, after that what happened? The structural proteins assembled with the negative mRNA. So we take the negative mRNA as well as we take the structural proteins. So the structural proteins are around them. So let's say these are the structural proteins. So the structural proteins are around them, and we can get incomplete viruses. Now after that, these incomplete viruses are transmitted to the Golgi bodies. First of all, it is transmitted to the endoplasmic reticulum, and after that means endoplasmic reticulum, and after that it is transmitted to the Golgi bodies. From the Golgi bodies they can get their envelope everything, and then it is eliminated, or they can although it is endocytosed, so they rupture the cell, or they rupture the neurons. So neurons was ruptured, and then many viruses are released from their body. So let's say these are the progeny viruses. So mature, then each viruses are get matured, and they are ready to infect the other neuron. Okay, so this is the life cycle, or this is the pathogenesis of each rabies viruses. Okay, now as we talked that it is usually caused by the rabid dog, it is usually present at the saliva of the rabid dog. Okay, it is present at the saliva of the rabid dog. Now that's about the pathogenesis, and that's about the epidemiology and everything. Now As the pathogenesis and life cycle is done, after some time we get the sign and symptom of each diseases. Okay, but usually the sign and symptom are appeared or it depend upon on the incubation period of the rabies viruses. So if we see the incubation period of the rabies viruses, it depends upon on the site of infection. Now, on the side of the bite, if the rabid dog bite on the leg, so the incubation period may be long, or it may be lead to three months. Okay, so if the dog is bitten on the leg portion, so then it may be the incubation period of this rabies virus may be to 3 months. Now, if the rabid dog bite the person at the neck portion, so we better know that our face and neck portion, all these area are rich with nerves. So definitely it will be transmitted to the brain very fastly as compared to the leg portion. So then the incubation period will be very low, or we can say it may be 6 days, 6 to 8 days. Not this is not the exact value, but in some books they write 6 to 8 days, then the sign and symptoms may appear. So this is the incubation period. Then once the incubation period done, then we get the sign and symptom of each diseases. Okay, now what kind of sign and symptom? Let me erase and let me start the sign and symptom. Sign and symptom. So when the rabies viruses cause the encephalitis, so due to this way, all of you better know that brain can control all the activity of our body. They can control every organ inside our body, like lungs, like liver, other organs as well. But remember, when they cause the first things of the sign and symptoms, in this case we have encephalitis. This is the first, mean inflammation of the brain is happen. So when you have inflammation of the brain, then usually we have what? We have paralysis. Paralysis. We have another sign and symptom is insomnia. Hydrophobia means the patient is fear with water. Patient have fear with water. Hydrophobia. We have another sign and symptom, we call that confusion. Paranoia. Hallucination. Anxiety and coma. These are the main sign and symptoms of the rabies viruses. Okay, although there are some other, but these are the main sign and symptom. Usually we have, if we talked about, there are 2 types of the rabies viruses. And according to them, we have the sign and symptom. So if we talked about the types of the rabies viruses, so we have usually 2 types. 2 types. In the first type we call that furious rabies. Second we call that paralytic rabies. Okay, so we have usually 20%, we have usually 80% we have furious rabies, and 20% we have this paralytic rabies. Okay, so in furious rabies we have hydrophobia, and in paralytic rabies we have usually paralyzed. Usually the patient is paralyzed. So these about the 2 types, the 2 main types of the rabies viruses. But 80% is rare in all country, in some of the countries as Asia, Africa. Okay, then we have the prevention of the rabies viruses, that how we can prevent ourselves from the rabies viruses. So we have what? We have the prevention. So we usually prevent ourselves from the rabies viruses that we should have to avoid from the rabid dog. We should have avoid from rabid dog, which we use as a pet. We also wash that area of our body which was bitten by the rabid dog for almost 15 minutes. Okay, so wash bitten area for 15 minutes with antiseptic or with usually with soap. Then we have another prevention in this case, that we should vaccinate our pet. If we use the dogs as a pet, we should vaccinate it. Okay, so vaccinate the pet, usually dog, vaccine. And another thing is that we should have to live vaccines are available for the prevention. So we have lived attenuated, try to remove the spelling mistake, attenuated vaccine. So live and attenuated vaccine are present for the rabies viruses. Okay, now these are the main prevention, that how we can prevent ourselves from the rabies viruses. Then we have the treatment of the rabies viruses. Okay, we have already we have this live inactivated vaccines are available, which we use as a treatment of the rabies virus, because rabies viruses are the viral disease, so there is no proper treatment of the rabies viruses. So if we want to prevent or if we want to treat ourselves with the rabies viruses, so we usually use this live inactivated vaccine. So first of all, you should have to take live inactivated vaccine when you are related with the rabid dogs. When you take the rabid dogs at your home, you should vaccinate yourself. Once you vaccinate yourself, and let's suppose so in the first treatment you should vaccinate yourself if there is rabid dog present with you. Then this is the first dose. The second dose is that you should, and then you should take another dose on the third day. Okay, again let me explain. The first treatment of the rabies viruses is that first of all you should vaccinate yourself. Whenever you are bitten or not bitten, you should have to vaccinate yourself. Once you vaccinate yourself, then after 3 days you again vaccinate yourself. Then you may be protected from the rabies viruses. Now, the second way of the treatment of the rabies viruses is that we have, okay, now if you are not vaccinated first, and let's suppose the dog bitten you, then you should take 4 doses, okay? You should take 4 doses. Now, in the first case, we talked that we should take 2 doses if we bitten or not bitten by a dog. Then we should take 2 doses. First of all, we should vaccinate ourselves, and after 3 days, then we should vaccinate. Let's suppose at that case, and at the second case, if you were exposed to the dogs, and let's suppose the dogs bitten to you, and after that you should take 4 doses, and in 4 doses you should take first dose right away, or on the right time, or on that time when you was bitten. After that, after the first day of the third day of the first dose, then you should take another dose. Okay, so 4 dose in the first dose is right away. Second dose you should take on the 3rd day. Then third dose you should take after the 7 days. And then 4th dose you should take it after the 14 days. Okay, then maybe there's a chances that you may be treated yourself from the rabies viruses. This is the 2 cases of the treatment from rabies viruses, that how you will treat yourself from the rabies viruses. So that's about the rabies viruses. Now another most important thing that I forget to mention early, that is about the diagnosis of the rabies viruses. That how we know that we have the rabies viruses. We can diagnose ourselves from the rabies viruses through each sign and symptom, but we have also some tests we should perform when we have these signs and symptoms. We should perform the test, and we have two types of the diagnosis. When you are alive, let's suppose, if we talked about the diagnosis of the rabies viruses, diagnosis. So usually we perform two tests for the rabies viruses. The first test we call that immunofluorescent test, and the second test we usually performed fluorescence antibody test. Okay, if we talked about the first immunofluorescent test, so in this case we take the sample from the patient. Usually we take the sample from the saliva, we take the sample from the blood, serum, or we take the sample from the CNS, central nervous system fluid. We take the sample from each area. After that, we have a test we usually call the immunofluorescence. In this case, what actually we do, we provide the antibody. Okay, so a lot of antibody we provided it, and we usually also provide our sample. So inside a sample, if we have the antigen, means rabies viruses, if we have, so each antigen will bind it. Okay, due to which a fluorescence or a signal is arises. So fluorescence maybe arises. Now each fluorescence or signals can show that your antibody is activated against each viruses, and they can show that yes, you have rabies viruses. Now this test usually we perform at live case, when the patient is survived or when the patient is lived. So usually then we perform this one test. This test we usually perform at death case. Usually we not use this test at all the time, but when the medical student or when the usually researchers or when the we can say that the CIG teams, all these teams or forensic lab, they usually perform each test. In this case, you know, they can take the dead body and they can usually target the brain area. So they see the brain. Inside the brain, or they usually take the tissue from the brain. Tissue from the brain. Okay. Once they take the tissue from the brain, then they usually detect each tissue with antibody. So they treat the tissue with antibody. Now after that, they will after that they see some negri bodies. Negri bodies. Now each negri bodies can show the identification of the rabies viruses. Okay, rabies viruses. So that's the two main tests or diagnosis through which we that's the two main tests through which we can diagnose the rabies viruses. Okay, so that's all about the rabies viruses. It is actually the short lecture on the rabies viruses, not a detailed lecture. This is actually the overview of the rabies viruses, and I hope that you will get that how actually the rabies viruses, what are the rabies viruses, how it is caused, how it is transmitted, everything about. Okay, but a little. Okay, so I hope you get the idea about the rabies viruses, and if you get the idea and if you understand, so then like it. Or if you have any kind of question, then you should write it into a comment. I will give you answer. So you can also share this lecture with your friends, and if you have need more diseases lectures, so you can subscribe my channel to get more lectures on the medical microbiology or the pathogenesis of every disease or the diseases lectures. Okay, thank you so much for watching this one video.